Hello, good morning, everyone. Thanks for being here. It's a pleasure to have you here, students and faculty from ENES Juriquilla, which is a campus from the UNAM, which is in Querétaro, 2-hour drive from Mexico City. And also it's a pleasure to have here students and faculty from Manchester Metropolitan University in the first talk of the Binational Neuroscience Seminar that we're starting together in form of future collaborations, hopefully between both institutions. So before we actually start, I just want to thank Doctora Araida Hidalgo for putting this thing together to trying to foster collaboration between both institutions, the Manchester Metropolitan University and ENES Juriquilla. Thank you very much, Araida. We hope this is the first part of a series of talks that we're going to have, basically starting in neuroscience, but hopefully in other areas in joint efforts between both universities. And also we want to thank Ana Elena González Treviño, which is in charge of the Center for Mexican Studies of UNAM in the UK. Thanks a lot, Ana Elena, for all your help and support in putting this program together. And as I was saying, we just hope this is a start of a good and productive collaboration between both institutions. And since we have a short time today, we're going to have two talks. The first one is Minds to Molecules, a whistle-stop tour through neuroscience teaching and research at Manchester Met. And the talk is going to be by Lloyd Orton. The second talk is going to be by Doctora Magda Giordano. She's going to talk about hiding meanings, the neural and cognitive basis at Pragmatic Language. We're going to let them speak about 25 minutes, and then we have a chance for students to ask some questions, or faculty, whoever wants to do it. So use your chats in Facebook and YouTube or here at Zoom. And we want to welcome all of those that are following us at the different streaming content. And I just want to thank Lloyd Orton for being here. He's a senior lecturer in neurophysiology in the Department of Life Sciences at Manchester Metropolitan University. He's a program leader for the MSc Human Physiology. He's also path leader for the scientist training program in clinical science in neurophysiology. He also leads neuroscience focused units on the human bioscience and human physiology undergraduate degree. He has a PhD and a postdoctoral training in auditory neuroscience at Newcastle University under the supervision of Adrian Rees. And he has, since starting his career, contributing to teaching and research with a focus on sensory processing, particularly the auditory system, and he has ongoing research projects with the Center of Bioscience, including investigating the recently discovered link between hearing loss and increased risk of developing dementia. Thanks a lot, Lloyd, and it's all yours. Thank you.
Thank you very much for the opportunity to present. I'll just try and share my screen. And hear me okay?
Yes.
Yes.
Yeah.
And you see that laser pointer as well?
Yes.
So, okay, thank you very much for the opportunity to present and be the first speaker. Hopefully this is going to be the start of a really fruitful collaboration between our two institutions, and it's a real pleasure for me to introduce you to Neuroscience at Manchester Met. One of the things that this is quite a new thing for us to do, so one of the things I'm keen to foster is some sort of dialogue. And so I'm really keen for outside of the session to pick up any future conversations you wish to initiate. So you can either scan the QR code or go to this link here and provide some feedback on the session. I'll put these up at the end of the presentation so we can hopefully start an ongoing dialogue outside of these sessions. So as Raul very kindly introduced me to, my presentation is based around minds and molecules and covering the wide range of things that we perform in terms of neuroscience research at Manchester Met, from the very small molecular work up to whole body human neuroscience and neurophysiology. And first and foremost, I want to allay some of your fears. This is a picture of Manchester. We spread some very malicious rumours around the internet that Manchester is a wet, dark and gloomy place. But actually it's very beautiful, very sunny, one of the hottest places on earth. If you look outside my window, there's blue skies for miles and miles, and a radar will definitely confirm that Manchester has wonderful weather, and it is a perfect place for people who are born from a very hot climate to visit. So don't be put off by those rumours. Manchester is far nicer than we let other people know outside of these meetings. So what we'd like to try and do in this session is basically introduce some of the research that we do here in terms of neuroscience at Manchester Met, and then see how that feeds into the teaching at both undergraduate, postgraduate and PhD level, to give you more of an understanding of the way that we work and what we can potentially offer. And we really hope that we can build upon this collaboration to help shape a collective future. And one of the things I was seeking for when I was trying to look for links between Manchester and Mexico was a kind of a bridge beyond my love for Mexican cinema with Luis Buñuel and Alfonso Cuarón, and I found a potential link between temples. So this temple, I'm sure you're all very familiar with, is where I was supposed to be in May, and thanks to COVID, about a week before, I was barred from flying out, and I've yet to visit your beautiful country. And whilst I hope one day to make it to Mexico City, a major priority for me is to make it to the Yucatan and Chichen Itza. So hopefully at some point I'll get an excuse to visit your university, and I can maybe fit a visit into one of your wonderful temples. We also have temples in Manchester. We have a very good pub called the Temple Bar, which I hope to welcome you to at some point. And we also have my favourite temple, which is Old Trafford. And you can see here the major canal that links Manchester with Salford towards the coast, which is how Manchester became one of the leading industrial cities in the late 18th century, and has sprung us into becoming one of the real centres of global investment in the modern day. And hopefully we can all unite around Javier Hernandez and the link between Manchester United as a thing that can unite us and bring us together beyond just pure neuroscience. But getting beyond that, I thought I'd tell you a little bit more about how our department sits within the local infrastructure and more globally. And we are, as you're aware, a more modern university. We've been around for a lot longer than 35 years, but we have 35 years of neurophysiology and neuroscience educational experience, and a growing number of specialist staff dedicated to delivering excellence in teaching related to the neurosciences. We've recently received investment in a new building that's due to open in 2023 for £125 million. And neuroscience is a key component of our vision for how we're going to take research forward and informing our teaching associated to that. And because of our long-standing history of both education and growing expertise in research, we've got excellent student satisfaction and graduate prospects, which I'll tell you a little bit more about as the talk progresses. One of the things we see as a key strength is that we have huge links with the local National Health Service, which is one of the largest health services in the world's first public health service, meaning that it's free at the point of care. And we have trained a plethora of physiologists across a range of specialisms, including cardiac, but particularly I'm involved in the training of neurophysiologists. These are the people that you go to if you potentially have suspected epilepsy, other forms of dementia, multiple sclerosis, ALS, etc. And we train the local practitioners right up to consultant level and across a wide network throughout England and Wales as well. So we've got huge clinical networks that allow us to bridge our basic research focus with a clinical focus as well, that allows us to bridge that translational mapping towards bringing new things, towards helping impactful changes in the world. Our research has grown in the past few years. Our government has an exercise every 5 or 6 years in which they appraise the research done at different institutions, and the two key components of them. Sorry, can you hear me?
Yes, go ahead.
Yes. Yeah.
The two components of that are our research grants, which we've received 3.2 million from a number of national and international funders over the past few years. I just saw one of our professors in the chat, Professor Dagans, who has funding from the UK Space Agency and the European Space Agency. And we have millions of pounds worth of funding from researchers. I found out yesterday that I've just been awarded an Alzheimer's Research UK grant to look at human brain bank tissues. And
Congratulations.
Yeah, thank you. They don't come around as often as you'd like, but when you do, I thought I'd brag about it in this talk. And so we have increasing numbers of these grant awards coming in associated to neuroscience. And we have the other thing that our government assesses, our research expertise, which is involved in the quality assessment of research. And we publish in top quality international journals. So journals including Brain, Psychological Medicine, the Journal of Neuroscience, Neuron, eLife, Stroke, and the Lancet Neurology are all recent lead author publications across our new Neuroscience Research Centre. So we've got a growing strength across a number of areas that we hope will appeal to a broad number of neuroscientists at your institution. And I thought I would kind of break down some of the experts that we've got here in the time available and talk about what their areas of expertise are. So you get a flavour beyond just my research as to what we do here. And the leader of our Centre for Bioscience is Professor Tristan McKay. And he's got a particular interest in modelling metabolic deficits in Batten's disease. This is a condition which is sometimes referred to as a dementia of childhood. It's a familial linked genetic disease whereby children inherit a range of lysosomal storage diseases which manifest in a heterogeneous way, but they lead to very profound and negative consequences. And Professor McKay is part of this European consortium called BATCure, and they hold the largest repository of Batten's disease-induced pluripotent stem cells worldwide. So mainly focusing on cell culture models of human pathology here. One of the beautiful things about iPSCs is that you can then induce human mutants, so human familial genes, for instance, that lead to Batten's disease, such as this one here, and you can knock them in, and then you can perform various chemical modulations to induce things, different cell types. You can induce neuroprogenitor cells. You can also induce neurons, glia, such as astroglia, astrocytes, and microglia, based on various neurochemical factors. And then Tristan performs and heads a group leading a whole range of modern molecular biological techniques, so proteomics, metabolic profiling, and as well as putting these in vivo for testing, and has led to discoveries including novel nuclear phenotypes in this disease, which is very early days in terms of helping these patients, but at least it's starting to characterize them. And importantly, it's got huge links for drug profiling and for potential future interventions that may help these patients. And this has been supported by Horizon 2020 funding. Tristan's also leading a cross-disciplinary piece of work in modeling, using his models to help develop a novel blood-brain barrier in vitro model, a cell culture polycellular model, whereby you can therefore screen for drugs in conditions associated to other aspects of blood-brain barrier pathology as well. Our new neuroscience group sits within the Centre for Bioscience. Within that, we also have three other groups. We have a cardiovascular group, a microbiology group, and we also have a aging and lifelong health group. The neuroscience group is new and builds upon the dozen or so researchers that we now have, and is now headed by Professor Sonia Correa-Muller, and she is interested in molecular mechanisms regulating synaptic plasticity and cognition. And she works a lot with genetically modified animal models, such as mouse models, and is particularly interested in hippocampal function. So classic approaches such as the Barnes maze here, the Barnes open field test here. So where you can use allocentric external cues to the mouse. Mice tend to like being in dark environments. So you put them in a brightly lit circular table with all these little holes around the perimeter. With one very dark box they can escape into. And mice typically want to escape into that as fast as they can. And you can see that when they put a wild type mouse in this environment on day one, they explore, these blue lines show exploratory behaviors around that until it finds the escape hole. And you can see by day 5 and 15, this is performed much more efficiently. And a gene that's been modulated here, the ARC early gene, immediate early oncogene, has been modulated. It shows similar performance in learning this task. But the animals have a real issue when the task is reversed, so that the hole here, as a top-down view, you can see that the hole is reversed from here to here. So the mouse now has to relearn where to go using these exogenous cues. And in a couple of days, the wild-type mice get very good at this. But these knockout mice have a real impediment to understanding how to perform this task. So they initially go to where the initial hole was, but they then make a rapid movement around the perimeter. They don't like the open field, so they stay near the edge to escape. So impairments to these animals' behavior can then be associated to this genetic modulation. And she can also look at things in electrophysiological paradigms. So here in the hippocampus, stimulating the Schaefer collaterals, which innervate CA1, doing field potential recordings. And in vitro, we can also do single cell work as well and target other parts of the hippocampal formation, including subiculum and entorhinal cortices as well. And she's received funding from Wellcome Trust and AstraZeneca. A relatively recent appointment who's working with myself and Professor Correa Muller is Virginia Hawkins here. Dr. Hawkins has a real interest in ATP and purinergic signaling and potassium channels, in particular inwardly rectifying potassium channels and their signaling at brainstem astrocytes. She's got a real interest in the RTN nucleus here, down in the really near the spinal cord, down in the brain stem, and she can perform patch clamp recordings and look for electrophysiological correlates of changes associated to mutations. She's working with me on a transgenic rat model that we have of Alzheimer's disease, as well as numerous mouse models. And also she performs things like immunohistochemistry and cellular imaging, single cell qPCR and RNA-seq using our new Illumina system. And as well as another technique called plethysmyography, which is a technique whereby you can get measures of whole body respiratory function. And on the masters that I lead, she's had a project student this year who's identified in astrocytic subtypes in this mouse model modulations to the ACE2 receptor, which is the receptor for COVID-19, the molecule that the virus apparently binds to, and which leads to potentiation of signaling within this network, which may be one candidate through which people at risk, say with dementia, are potentially dying at higher rates due to COVID. And so this is an active area of real innovative research here. And here's Ginny at the patch clamp rig, looking down a microscope, but here you can also see a camera where she can watch the cells live and then patch onto them and record the electrophysiological characteristics of their cellular phenotypes as well. This is Dr. Nicola Ray. Dr. Ray is primarily interested in movement disorders and particularly in Parkinson's disease. So Parkinson's disease is classically characterized as having a triad of motor deficits, as well as later cognitive and dementia-related deficits, and increasingly recognized as having other extra Parkinsonian symptomatology. And here she is, looking very snazzy in her EEG cap. This EEG cap here, you can see each of these points allows an electrode to contact the head in a classic kind of 10-20 orientation, and allow spatial localization to within a few centimeters of where electrophysiological signals are generated within the brain. One of the classic things that EEG does is identify ictal and epileptogenic signals from the cortex in epilepsy classically. But importantly, it also has artifacts when we have movement, and the signals are modulated, whether it's a neural substrate or say a motor substrate, muscles in the neck and the face moving. And so she's got this new system whereby she can correct for that and actually get signals while people are actually moving, which is a really new innovative approach in EEG. And so therefore she can ask people to perform tasks whilst recording from the head and try to look for cortical correlates of those potential motor functions and pathologies. She combines this with functional MRI, and you can see here's a parasagittal, mid-sagittal section through the brain, and you can see this is the plane of section here in the coronal plane, and she can identify these basal forebrain cholinergic subnuclei, and was able recently to demonstrate that these are associated with an increased risk of mild cognitive impairment. So those with deficits in this cholinergic brainstem region, which are classically associated with damage in Alzheimer's disease, appear to be important also in Parkinson's disease, that these people have mild cognitive impairment and early stages of dementia-like symptomatology far more common and far sooner than those without. So this is potentially looking for early biomarkers and is being extended to other brainstem nuclei as well. So here we're zoomed in the sagittal plane again. You can see the cerebellum here, and here's the pons going into, this is the pons into the midbrain. And you can see that she can identify individual neuromodulatory nuclei deep in the brainstem here and try and see whether they can be looked at in early Parkinson's disease or indeed in the prodromal stages during just healthy aging to see if these can be potential new signatures that could be used to help screen populations. And she's received extensive external funding from Wellcome as well to support this work. We also have two professors who collaborate extensively here, so Professor Mark Slevin and Professor Jurek Krupinski, and they're interested in risk factors and biomarkers of stroke and neurovascular disease. particularly things around genetics and epigenetic risk factors. This is work done in conjunction with Professor Murgatroyd, who's going to be speaking next week, as well as other aspects of neurovascular disease. And because Professor Krapinski is also a consultant neurologist, spends a lot of his time at University of Barcelona, he has a group there where he can access lots of brain samples from stroke patients who sadly passed away, but also living patients as well. And this is combined with a lot of Mark's expertise relating that to both animal work and cellular work as well. So looking at things like C-reactive protein in vitro models, as well as in vivo and ex vivo models, and a very extensive research repertoire between the two of them. Oh, is my screen frozen? I appear to have frozen. There we are. And then finally, I thought I'd tell you a little bit about my work. So I'm really interested in auditory processing in health, but I've also got an interest in age-related hearing loss, something that all of us, if we're fortunate enough to live long enough, every single one of us will acquire some form of age-related hearing loss. And particularly interesting are novel aspects of microglial and astrocytic modulation, things like this confocal micrograph that shows microglia in green and astrocytes in magenta here, lining a blood vessel and lining the perimeter of the brain. And we can show how they are dysfunctional in age-related hearing loss and potentially lead to novel observations as the link towards increased dementia risk. So the recent Lancet study in 2017 identified as the leading cause of preventable dementia cases is due to hearing loss in middle age. And we don't really have any good models for why that is. So I've been working for a number of years on these animal models, and particularly focused on this transgenic rat model, which both contains age-related hearing loss and human familial Alzheimer's disease genes. And we're able to look, therefore, at the interaction of hearing loss and Alzheimer's genes. And we've identified novel aspects of pathology, including presynaptic mitochondrial dysfunction and gliosis and reactive pathogenic responses to decreased GABAergic inhibition. And we're using these to kind of tractably investigate novel links between hearing loss and dementia. I also use techniques very similar to Sonia and Virginia, where looking at electrophysiology in vivo and in vitro, as well as mitochondrial biogenesis and other aspects of molecular biology and biochemistry. I've also got collaborations where we can do PET imaging and micro MRI as well. One of the great things about rat models of disease is that they're large enough to allow us to do things like single cell identification of PET images and modulation in fMRI as well. And we can drive that with sound stimuli. So we can really tease apart some of the things in the living animal, which is really great for translational work. More broadly then, we've had this large extensive investment in neuroscience and in our faculty more broadly. So as I mentioned, we've got £125 million building coming in 2023, as well as a brand new fMRI that was ordered 3 days ago. We've also got a new Illumina sequencer that allows us to do next-gen sequencing at single gene level, right the way down to single alleles. So huge bioinformatics approach is taken now in a lot of our work, as well as we've just invested this summer in a new flow cytometer that allows us to cell sort and new qPCR machines as well. For any of you that are brave enough to come to Manchester, hopefully you'll be even braver and come to our BEAM lab here, where we can model things like motor pathology at height, which is really helpful for modeling age-related fear about walking in young, healthy people. Or if any of you are climbers or just generally like an adrenaline rush, we're always looking for fresh participants. You're allowed to back out at any time, but maybe that'll be for only the bravest of you. And all this research feeds into our teaching portfolio extensively. So we have a number of degrees here. So we have degrees in biosciences and physiology, as well as the MSc Human Physiology that I lead. And as I alluded to earlier, undergraduate, postgraduate taught and consultant level neurophysiology degrees. And we have neuroscience across each of these in multiple places. And these are a bunch of my summer student neuroscientists from a couple of years ago now. And you can see we've got a real good bunch of keen students. These students all came in for 8 weeks, and they were only paid with one trip to Nando's at the end of the summer. So coming in and working hard and really keen to get involved in neuroscience research, in addition to their their learning at the educational establishment that we have here. So it's a great mix of people that we have, and we hope you're keen to come and join us. So we have multiple neuroscience units that are explicit in their reference to neuroscience. I lead this neuroscience and pharmacology unit in the second year, where we take students from a crash course from receptor right up to whole body in pathology and basic physiology of the brain, as well as aspects of pharmacology as well. We have a new advanced cellular and human neuroscience unit that I contribute to, which is very much research focused, aligned to a lot of the work that researchers like myself, Dr. Hawkins and Professor Correa Muller contribute to the department. And also I lead an advanced aspects of neuroscience unit, which is purely research and practical based, and is on our MSc in human physiology. And this is all around things that basically that are purely research informed and research led. So we have multiple ways at which we can add to the educational experience of neuroscience across our degrees. So why might you want to come and study neuroscience at Manchester Met if you're a student? Well, everything is research driven and it's taught by researchers working at the cutting edge. We have unique experiences that can only be delivered by our neuroscientists, and every single neuroscientist I've talked to you about today does a lot of teaching to give back to the department and give back to our students. And we also have a growing international reputation for neuroscience excellence, and we have very, very good real-world impacts of this, so we can show how this benefits society as well. Growing investment in our facilities, and a number of levels of expertise at which you may wish to collaborate. So hopefully we can collaborate across multiple domains and multiple levels of approach to neuroscience. Where do our students go on to? Well, this is Damilola. She was one of the students on the MSc Human Physiology when it first started 3 years ago, when I first set it up. Here she is working on one of our anatomage tables. This is where it allows you to dissect the human body without having to have a cadaver, which is really handy for teaching as well as clinical work. And she's now gone on to become a medical doctor. So she's now training as a graduate entry medicine here. And this is a quote from her talking about how great an experience she had. We also have a number of students that take the scientist training program after working here and studying here and become physiologists, particularly in the local health service. We have a number of students that go on to be research scientists, a number of neuroscientists that then go on to do further postdoctoral and academic work. And we've got a number of students as well from the MSc Human Physiology who've gone on to be project coordinators in medical communications companies. This is a real growing industry in Manchester, as well as medical writers. And we have a massive industry now in medical writing. This is an increasing and expanding field, which Manchester is a really attractive proposition to companies that previously were based in London. They're now being priced out of London, not because of the bosses, but because they can't attract young people because they can't afford to live in London. So a lot of them are moving their companies to Manchester now, which is kind of seen as the second best city in the UK by many people. And these fields are really burgeoning. So you can build upon that, and we use our networks to really build upon your studies here. So there's a huge number of possibilities, whether you're studying with our academics in a lecture theatre, but also this is the Friday Five-a-Side football team that I put together 4 or 5 years ago now. It's a mixed gender five-a-side game that we have every Friday night. Everyone's welcome. And this really shows our international portfolio of people. So we've got Matteo here is Italian, York here is Chinese, Guy here is from Congo, Fabien is French, Lorna is Italian, Lorna is Irish, Mia is Bulgarian. So you can see we've got a really international flavour. Saraga here is from Malta. And it's a really welcoming, diverse community that we're really keen to take advantage of in Manchester. It's one of the great aspects of Manchester. It's so welcoming to so many different people, and there's so many opportunities, and no matter your background, and you can really take advantage of that at Manchester Met. So that's about all I had time for today. I'm sorry I didn't go into much more detail about the research, but I felt a kind of broader overview may have been more helpful for you all. And so, yeah, I'm happy to take any questions, and I really looked forward to reading any feedback you may have via the MS forms after this session. And if you need me later on, this is me. I'll be hanging out on the sofa with my 2 cavapoos. So thank you very much for your attention, and any questions?
Thanks a lot, Wade, for your presentation. A very nice overview of all the research that is done at the university. I don't know if the students have any questions. Nobody's, there are a lot of possibilities if you guys want to follow a possibility path in research there, or maybe even thinking about doing a stay for a semester or something. Is that possible?
Sorry, could you say that again?
For example, the students who go there for a semester, will that be possible?
Yeah, so we've got an established Erasmus program with Europe, which we could easily model into our current approach to studying. So we've got experience of students from North America, for instance, have come in from that. I've had multiple students on the second year neuroscience unit that I've led, and they've spent time studying neuroscience with me as part of their undergraduate studies. So that's very much possible, and the credit bearing, credit sharing arrangements would be easily transferable, I think. Would need some discussion, but that's definitely an opportunity.
Yeah, that's something that we probably we want to pursue as an institutions, to try to, with the help of Ana Elena, and try to have some form of collaboration so the students go back and forth, from both universities. That would be nice.
Yes, we're working towards that. Yes, I'm happy to say yes. Mm-hmm.
Yeah, that would be fabulous. Yeah, I think that'd be a really good opportunity for students. Yeah.
There are a couple of questions for you about your research. Is the loss of hearing that leads to cognitive deficit, or is it hearing loss a manifestation of early cognitive decline?
Yeah, that's an excellent question. And the answer is we don't know. We certainly know that, for instance, people with early hearing loss, say congenital hearing loss, who develop language skills other than auditory, so sign language, for instance, they have much better cognitive function later in life. Some of the communication skills there that are attained through other means mean that you don't just get dementia if you've got hearing loss. It's typically midlife and later life hearing loss. So it's a midlife risk factor. But furthermore, yes, there's also another issue, which is the tools that we use to diagnose both conditions are really fallible, particularly audiological screening. So raised thresholds is a really poor measure, actually, of multiple aspects of auditory function. So when you first lose your hearing loss in older age, most people don't notice that they got a raised threshold at 4 kilohertz. They notice they can't talk very easily in a noisy backgrounded environment, and the signal to noise ratio degrades there. Similarly with using MCI and prodromal measures that lead towards dementia diagnoses, they're actually really full of false positives. So I've been doing a project on dementia with Lewy bodies, which by some measures in life is diagnosed at about 4% of dementia cases, while Alzheimer's is about 62%. You look at the neuropathological evaluation, and a huge number of those Alzheimer's cases are actually dementia with Lewy bodies, and they've been misdiagnosed. Same with Parkinson's. So these biomarkers and neurodiagnostic techniques need to be refined and developed. And that's why the animal models are so useful to try and give us indications, because at the human level, it's so hard at the moment to unambiguously say what you have. And people like a label, people want a diagnosis if they know something's wrong, so doctors are more likely to give that, but there are false positives at both sides of that. So yeah, there's lots of things to tease out there.
Okay, a couple of more questions. The last two questions, because of the time constraints, but there will be a lot of opportunities to have more questions. You already gave your email. Karen Delgado is asking if you guys study the plasticity of organization plasticity in the spinal cord during the recovery of an irreversible accident?
Not that I'm aware of. We have a very large musculoskeletal group at Manchester Met who are interested in a range of different motor conditions, so there would be interest in that. Professor Ian Lorem, you may want to look up his work on postural control, for instance. He works on that Beam Lab. There are a number of experts in that, but they tend to work on a non-human primate model, say at Oxford or Newcastle. I know people like Andy Jackson at Newcastle who work on that project, but there's nothing really Manchester Met. Spinal cord's really tricky in rodents. They recover far too well. They recover much better than primates do. So most of the animal work has to be non-human primate work.
Okay, and the last question, el Dr. Alberto Prado, aside from rats, what other animal models you guys use at Metropolitan University?
Yes, we use a number of models. So in age-related hearing loss, I use guinea pigs, ferrets, and a range of different mouse models. We have access to APP-SWI, triple transgenic, and PS1 mice as well. Dr. Hawkins works with Stuart Allen on those. And the main model is the transgenic rat model, the TGF344AD rat model we have. So we have a number of different available models for various things. as well as we can. So Dr. Craig-Muller, Professor Craig-Muller, she actually used genetic knock-ins as well, so she can actually use that with her collaborators. So there's a lot of different potential models that we have.
Okay, thanks a lot. So we're going to go to the next presentation, or the second presentation by Dr. Magda Giordano. The title is Hiding Meanings, the Neural and Cognitive Basis of Pragmatic Language. Dr. Giordano is a full professor at the Institute of Neurology here at the same campus, and actually she's now the academic secretary of Inés Juriquilla. She studied psychology in Mexico, and then she got her master's of science in experimental psychology and PhD in experimental psychology in the United States, working on the functional effects of neural transplants. He also worked as a Fogarty Fellow at the National Institutes of Mental Health, and later returned to Mexico and got involved in UNAM. She's been studying for a long time the role of basal ganglia in the control of behavior. She then, basically in animal models, then she switched a little bit to study the neurotoxic effects of environmental toxicants, such as heavy metals, on basal ganglia function. And more recently, and that's what she's going to tell us about, her plastic brain changed to study the neural and cognitive basis of pragmatic language. So thanks a lot, Dr. Giordano, and please go ahead.
Thank you very much. And I'm going to switch gears a little bit, and I'm going to tell you about what we've been doing in the last few years. We, as a cognitive in this area, we're talking about cognitive neuroscience, and our job as cognitive neuroscientists is try to understand how the brain does its work. We try to study human abilities, and so the number of techniques that we can use to do that are more limited than if we used an animal model. So I'm going to tell you about what approach we have used to study this particular kind of language, which we all use, but we sometimes are not aware of it. And so what is pragmatic language? And we can find it in poetry. And I put two examples here. This is a popular song, and I will only read the first line. It says, "Ain't no sunshine when she's gone." And then, another poem from a famous American poet. She says, "I felt a funeral in my head and mourners to and fro." And as you can probably understand, they're not saying exactly that is happening, but they are giving us a way to understand how they are feeling. In the first case, alone, without warmth, because somebody has left. And in the other case, not very happy, suffering some sort of mental disturbance in a particular moment. And we can understand that some poems are difficult, but we use this language in everyday language too. For example, we might say, "It's so hot in here." And we're not just talking about the temperature. If the window is shut, we're asking somebody to open it, but we're not asking that directly. And we will use proverbs very commonly in our language. I don't know in the UK, but in Mexico, we use them a lot. And I've given you here an example of all that glitters is not gold. And we also use irony or sarcasm. So you may enter your child's messy room and say, "Nice place you have here." So you're saying something other than what you're saying. You're communicating more than what you're saying. And these are different forms of pragmatic language that we'll talk a little bit more later. So how is pragmatics different from semantics? So semantics studies how words connect to things and attempts to establish if the relationship is accurate, true, or not. And around in the 1950s, around that time, linguists in the US started to question this notion about language being able to detect a truth. And they understood that language is more than this particular relationship to reality. So pragmatics is maybe defined, as I say here, as the ability to communicate by expressing and recognizing intentions of what it is said. So it is a study of speaker meaning, what I want to say. It is a study of contextual meaning, what do I mean in this particular context? And it is a study of how much more gets communicated than is said. And it is also the study of the expression of relative distance. There are things that since I do not live in Manchester, there are things that I may not be able to understand in a conversation because I do not have that closeness to that culture. So pragmatic forms include speech acts, which is what I told you about when you say it's very hot in here and you're asking somebody to do something. So speech acts means that you do something with your words. And we have some speech acts that are, you would say, authoritarian in a sense. When you get baptized, for example, somebody is doing something with their words. Or when you're named, you are doing something with your words. Also, when you're begging or where you're promising, you are doing something with your words. Those are speech acts. Metaphors, I showed you a couple examples. Those are easy, we'll all understand them. Idioms or proverbs, and irony and sarcasm are other forms. So this is an example of pragmatics in action, and I'm going to try and look at the time, which is, pragmatics can be found easily, I think, in polite conversation, in politeness. So this is from a book by Yule, and he gives us here an example. It says, how to get a pen from someone else. You can say something, or you can say nothing, but do something, like search in a bag. You can say something on record, or off the record. I forgot my pen. So you're not asking anybody to give you a pen. You're just saying, I forgot my pen. And then there's a face saving act, or bald on record, for example, give me a pen. A face saving act can be positive politeness. How about letting me use your pen? Or negative politeness. Could you lend me a pen? So we use this indirect language in common conversation. And many times it is to save face, or not to commit ourselves to a definite course of action. We don't know exactly what the other person intends. We don't know that person that well, but we still have to communicate with her or him. And we find these hidden ways to do that. So what we were wondering is, how is this type of language different in terms of its neural and cognitive underpinnings? So first, does it require additional cognitive processes, or does it rely just on the same language abilities that we have for semantics? And does it involve other brain areas than those involved in language? Does it recruit different areas of the brain? So I'm going to tell you just very briefly about some of the cognitive functions that we have explored, and we have tried to find if there is a correlation between understanding pragmatic language and these abilities. So one is called theory of mind, which is basically the ability to infer mental states in others. And that this is a function or a process that develops. You do not find it in young children. You find it around 4 years of age. And so, for example, that you know that somebody does not know what you know. That is theory of mind. Then there's a whole bunch of functions that are called executive functions that it's like a mixed bag of things. But some of them include working memory or cognitive control and goal-oriented behavior in general. So we wondered if these are needed in order for people to understand pragmatic language. And then, does it involve brain areas additional to those involved in language? And this is just a figure from a paper by Frederici, who's an expert in language. And I'm just showing you here Broca's area, which is the motor part of language, which is kind of in charge of speaking, and the auditory part, which you were talking about, which is Wernicke's areas, more in the temporal lobes. And these two areas are interconnected. And I'm just gonna I just want you to focus on the arcuate fasciculus, the different segments of the arcuate fasciculus, which connect Wernicke's region and Broca's region, and provide the basis for us to express ourselves in tandem with the auditory part. So those are like the two big main areas of language. And I'm just going to show you in this figure from Duncan. I just want to show you in the bottom figure, you'll see the areas that are involved in language. This is an example of one one individual. And then there's this other part that you can see in purple, which he calls the multiple demand system. And this multiple demand system, which would be the one that sustains those other kinds of executive functions I talked to you about. So you can see that they do not overlap. Why am I showing you this? Because this is our question. Does pragmatic language involve both networks? Does it involve language areas and also language areas involved in multiple demand tasks or functions? And this is another study from Diacek. On the bottom one, you can see in red the masks. The mean is to the left, and these are participant-specific masks. Areas that are involved in language, and on the top, areas that are involved in this multiple demand system that they found using MRI, functional MRI, using tasks like arithmetic and memory and other kinds of tasks. And these are just purely reading either sentences or words. And so what do our studies suggest up to now? The first thing we did is we did a meta-analysis that was done by Azalia and Elizabeth, and they looked at all the studies that had done this contrast, pragmatic language more than literal language. And we put them all together regardless of the pragmatic form, and we found this kind of distribution. I should remind you, these are means from a lot of people. And you can see that we have something similar to what I showed you in the image before. Language areas that seem to be involved, some frontal, inferior frontal activation, which are language areas, but a little bit more, especially in the middle of the cortex. So what we concluded from this is that these pragmatic language comprehensions involves a bilateral frontotemporal and medial prefrontal network. And we think that the interaction between these regions that are involved in language and areas related to social cognition, which are those areas are the ones that are in the middle of the brain, are independent of natural language, modality of stimulus, and writing system. So we checked if this was true for different natural languages and writing systems, because languages are, as you know, are very diverse. But we found that they did not make so much difference. So this first study showed us that this human language as a communicative tool involves both the semantic activation, integration, selection, and intention recognition, which is this part that is really relevant for pragmatic comprehension. So pragmatics is the capacity to communicate in a way that is fundamentally a matter of social cognition. And that is what this meta-analysis showed us that pragmatics, in addition to recruiting areas involved in language per se, are recruit areas involved in social cognition. And we have continued to work. This work is from Giovanna's master's and PhD, and she's looking for areas that are related to speech acts. Remember, I told you that speech acts are acts that we make or do with words. And what she found, which is interesting, is she found activation of a region that's called the precuneus, that it has been indicated as a part of what's called the intention recognition network. So we believe our studies, this study in particular, is congruent with this idea that speech acts involve the detection of intention in others. And another interesting finding from this study is that we found a significant correlation between mental flexibility and the changes in bold signal, which is the signal that we detect with functional magnetic resonance imaging, precisely in the precuneus. So this ability to switch from one way of thinking to a different way of thinking appears to be related to this change in activation. So that I told you, and since we're running out of time, I'm just going to show you a couple more examples. Another thing we've studied is metaphors. And what we found is this particular pattern of activation when we compared metaphors, more than literal language. So we found activation mainly, interestingly, in language areas, frontal areas, and also the middle that I showed you before. This part, the middle prefrontal cortex, and also parietal cortex. And this is another type of contrast. We just use different type of stimuli. In this case, we found, we did the contrast metaphor more than literal plus absurd. Absurd phrases for us were our control for metaphors because they share some characteristics. And we found the same finding. And also an interesting finding from Miguel Angel's work is that the change of bold signal in the paracingulate gyrus is related to theory of mind, to the scores that people have in theory of mind and empathy. Again, showing that these functions that are related to social cognition appear to be important for pragmatic language comprehension. And finally, I wanted just to tell you why pragmatics is important to study. So who does not understand pragmatics? Children. They take time. It develops. It's not that you immediately, when you learn to speak, you understand pragmatics. It takes time. One of my students, Edna Navarrete, is looking at how children develop in their comprehension of metaphors. Also, people with, there is a social communication disorder that has been in the statistical manual of mental disorders, has been, I think it's its 5th version, explained or described this type of disorder. And also people with schizophrenia, there are certain brain lesions that induce this type of changes. Even though people can speak, they can speak regularly, they understand speech, but they cannot understand pragmatic, indirect language or pragmatic language. So I told you this ability is acquired, it has its own development, and it can be affected without affecting language as we understand it per se. And there are also individual differences. And I don't know if you in the UK know this character, which is from a sitcom in the US, who is Sheldon. And he's a brilliant physicist who has no clue about what other people say and about the use of indirect language. And it's just an example of how not everybody has this ability. And what are the consequences when you do not have understanding for pragmatic language? Well, there's social isolation, interpersonal problems, and problems finding or keeping a job. And so in conclusion, I told you that pragmatics is the study of language in use, and that language does much more than communicate information. It communicates intentions and can build and destroy relationship, and it shapes behavior. So we're interested in knowing how the brain does this work. And I think this knowledge can help us understand each other better, find better ways to communicate with others, especially with those that do not understand pragmatic language. When we know that they're unable, then we communicate with them in a different way. And it could probably help us to communicate between cultures. I remember somebody coming to a poster to me at the Society for Neuroscience, and he was amazed. He says, this is what happens because he works with people from different cultures. And he says, it's not that they do not understand the language. They understand the language, but they still cannot speak to each other or cannot communicate with each other. So this is something that we have to be aware that it exists. And this is the group I've been working with some summer students. And this is our address, our email address, if you're interested in knowing more. Let me go back. And that would be it. Thank you very much for listening.
Thank you, Doctora Giordano. We have time for a couple of questions. If you can write it in the chat. There is one by Doctora Bedos.
Okay,
Could
yes.
you explain how mental flexibility was assessed in your study?
We started, I told that we have very limited, we have limited techniques to do this. So we rely on neuropsychological tests. In this particular test, there are some that are very common. For example, one is called the Wisconsin sorting card test. And we used a battery that was, I don't know, how do you say, validated in Mexican population that uses a test that is something like that. For example, so you start a card game and you put down some cards and there's a rule. So when, for example, the form changes, the rule is broken, you have to say that there's a mistake. Then you change the rule again and now it's the color. So the person has to understand that the rule has changed. That is how we assess mental flexibility, which is, you know, supposed to be a prefrontal controlled function. And
there's another question by Dr. Prado. How much pragmatic is cultural or a learned ability?
I think it is. I mean, it is learned, and I started to study language because I thought that this is because you learn language in a particular culture and it develops differently. You learn different ways or different things depending on the culture and the language because they go together. I mean, if you can it's difficult to understand a culture if you do not know the language. So I would say that they're interconnected. And I started studying language because I think it's a way in which culture changes the brain, you know, naturally. It's a plasticity thing. It's the language changes your brain, and we know that children are able to distinguish sounds from every language when they're very, very young. So they are able to distinguish between the U and the A in the French and the Ha and the Ha in Chinese, that we cannot do that anymore. No. Because we lose those abilities because we're immersed in a culture. So that's one thing, that in terms of semantics and in terms of pragmatics, by force, there is no other way. It is embedded in the culture. How you say things, no, in the UK, how's the weather? No, this is well, that's the typical thing they say, that the British say when they don't know anything else to say. And we have other ways in Mexico to speak to each other, to communicate. And I think that's the main difference. No? Pragmatic language is used to communicate with others. Was that clear? I hope. And
we have another set of questions. Somebody asked, I don't know the source, do MRI scans of people with autism spectrum disorder look different to people who do not have the disorder?
That's one of the studies we have had to stop because of the COVID thing. One of my students, Elizabeth Valles, has exactly that project, and she's looking at irony comprehension in Asperger, the population with Asperger, trying exactly to do that, because you know, that would be a proof of concept. If we can see that their brain engages differently than the brain of a neurotypical person, it would tell us something about how this language is comprehended. But we stopped it, so we don't know yet. We think there is some information from other studies, a few, that do suggest that it looks different. And who do not, yeah, that's right.
Okay.
And also there's a lot of studies about deficits in pragmatic comprehension with different kinds of brain injuries. There are studies. We don't work on that population, but there is a group several groups that work on that, and they do have differences. Can you deliver auditory spoken? Yes, we can, and we do that. We do that. I'm going to read it because you're not reading it. Can you deliver auditory spoken stimuli in the fMRI language paradigms? And if so, how do you control for the background noise levels? We do. Actually, the irony experiment and also the experiment with children uses auditory stimuli. And we have headphones. And so there are different ways of doing that, and we use headphones that allow us to deliver the auditory stimuli in spite of the background noise, and it works. I mean, we've tested it already in small groups because the COVID stopped our work, but we do that, and we chose to do that in irony, and we chose to do that for children because they cannot all read, very well or at the same level.
Thanks. That was Lloyd Orton who made that question. And the last question, because of time, Doctora, is it known that rhythm is important for speech and understanding language? Do you know if there are differences in the brain areas where rhythm is processed in people with problems understanding pragmatic language?
I don't know, but we know that prosody, which is, how you speak, is important, for example, for the understanding of irony. So I don't know about the rhythm itself, but the way you say things, the prosody, has a lot to do with pragmatics. And that's what Elizabeth is studying. She wants to have neutral, prosody that is flat, you would say, and prosody with an ironic melody, let's say, and to see if that makes a difference. It makes a difference behaviorally. People do not recognize irony if it's flat. Some people do not recognize it if it's on a page, But in regular communication, to understand irony, prosody makes a big difference. So, but I do not know if the rhythm per se is a factor.
Well, thanks a lot. That will be all. We just covered 1 hour. Just a reminder that on November 26th, we're going to have another couple of talks, and on December 3rd as well. And we just want to thank to all the students and faculty that join us today here at the Zoom session and also by Facebook and YouTube. And as I said, we all said at the beginning, we hope this is the starting of a fruitful collaboration in the future between Manchester Metropolitan University and Enes Juriquilla. Thank you all and see you soon. And like Debbie already says, use a mask. Don't forget your mask. And for the Enes Juriquilla, les recuerdo que a las 11 hay una plática también muy interesante, Relaciones de Género y Conflicto en África, por la doctora Fatuma Ahmed Ali. Esperamos verlos allá si pueden. Thanks again and see you soon.
Bye.
Bye.